Good morning, ladies and gentlemen.
Is it on?
Glad to see you all weathered the cold weather this morning and made it here successfully. Can you hear me back there? Good. So this morning, the title of this session is Reproducibility Within and Across Experiments. And to kick this session off, we're delighted to have Professor Andrew Macpherson from the University Hospital at Bern, Switzerland, give us the keynote this morning, which is creating stabilized and defined microbiomes in laboratory animals. I'd like you to all welcome Andrew to this meeting. Thank you. Andrew?
Thank you. Thank you very much indeed. I would like to thank Jim and the organizers for inviting me. And it's a fabulous meeting and with some very distinguished colleagues that are speaking. My title is very ambitious indeed, creating stabilized and defined microbiomes in laboratory mice. It was given to me, and I will let you I will let you decide at the end how far we can go along this particular trajectory. So the starting point is, of course, that if we think of the 3 R's of ethical animal experimentation to reduce, refine, and replace, at least for reducing and refining, we have to be able to try and control what the different factors are in terms of the variance. And this is clear that there is a major component of variance that is due to the associated microbiota, not just bacteria, but other microbes as well, as you've heard earlier in the meeting. And this, I think, is probably the hardest aspect of animal husbandry to control or standardize. And it's also true that the biology of this is not understood, which makes it harder to try and get a grip on standardization. And the consequence is that it has an impact on our ability to make robust measurements, to look at the underlying biology, to design suitable controls in our experiments, and to reduce the number of animals. So if we think that the phenotypic variance depends on the germline genetics, the environmental microbiota, and our experimental manipulations, of course, the history of animal husbandry has been hugely advanced by inbreeding to reduce the germline variance as much as possible, leaving the phenotypic variance to be predominantly the environmental microbiota and the experimental manipulations. So the subject of my talk today is to see how far we can go in reducing this environmental variance through the microbiota through notobiology, and of course it can't be reduced to 0. So another justification for animal experimentation, of course, is that it is biomedically important, and of course it is familiar to everybody in the audience that our bodies and those of all animals, of all higher organisms, are heavily colonized with environmental microbes. And this colonization reaches the highest density of microbes known on the planet in the lower intestine of 10 to the 12 organism per mil of contents. And the numbers are approaching astronomic proportions with the large number of microbiome genes, which contribute a very large part of our metabolism as host microbial superorganisms, and a comparable number of non-pathogenic environmental microorganisms to eukaryotic cells. And the relevance, of course, is strengthened by, both in animal models and in human studies, the demonstration that the underlying biology impacts many important and prevalent diseases. So just to show where we are heading for, the first thing I think is to look briefly at the degree of diversity between different human individuals. And these are 5 different subjects that were sequentially sampled over 6 weeks. It shows a 16S at genus level. And you can see that whilst the individuals, so feces and skin, so the first thing is that, as you would expect from many publications, that the feces and the skin are different, but the feces of each individual are reasonably consistent across this particular time, but they're different from each other. And that is the first point, that one microbiome cannot possibly characterize a species. The second starting point is that there are extremely strong effects, and the reason that we know this robustly is from the development of germ-free animals and the comparison of those animals before and after colonization with non-pathogenic microbes. So you can see here, this is an old experiment where black 6 mice have been either maintained germ-free or studied 21 days after the addition of a sentinel animal with a SPF microbiota containing approximately 70 OTUs. And you can see here the induction of IgA, the main secretory immunoglobulin, increase in the size and number of germinal centers of the Peyer's patches. And there are many other changes in the mucosa, but the point is that if one looks at all organ systems, essentially this sort of experiment comparing germ-free and colonized shows that the effects of the non-pathogenic microbiota extend essentially to all body systems. And this is the kind of technology for germ-free animals, these flexible film isolators with ports allowing sterilized food, water, bedding to be introduced, and then manipulation through the gloves. A second factor is that these that the effect of the microbes, of course, is due not just to the biomass of the microbes that are reasonably well confined at the surfaces of the intestine and at other body surfaces, but the effects of the microbes extend far beyond their live biomasses because of the penetration, the rather promiscuous penetration of microbial molecules all around the body. So this starting point leads us to consider the different types of experimental models that we might look at. And I've divided them here into top-down studies and bottom-up models. So my laboratory has been classically associated with these bottom-up models studying axenic germ-free animals or those with very simple microbiotas, monocolonizations, transitory colonizations. And of course, these only reveal part of the biology of the complex microbiotas that are studied in the top-down studies. They may have limited scope, they may omit important metabolic pathways, and they're mostly in mouse models. So these are disadvantages, whereas with the top-down studies, the big disadvantage that we're addressing today is imprecise definitions of microbial consortia, ambiguity in assigning effects to particular species within those consortia, and the biggest one of all, I guess, reproducibility issues, because we're in danger of having a decade of serendipitous results that can simply be ascribed to the fact that the experiments are being done in a different vivaria from the one that was originally publishing. So the bottom-up models also are more handleable for molecular mechanisms, whereas the top-down studies are more representative of a natural situation, although I shall come to the wiggle room of that in just a moment. So the principles of using inbred animals, and I'd like to draw a contrast here, the principles of using inbred animals are well known, that there is less inherent phenotypic variability that the experiments are much easier to power with smaller animal numbers than using outbred strains, and that the loss of genetic traits can be compensated by using different strains in a factorial design, all of which are properly powered. In other words, this isn't one, I think everybody would be comfortable with the idea that there is no one inbred strain that can be made to read out the different traits that we want to look at in terms of experimentation. But it is possible by using different strains to compensate this very considerably. I shall come back to that with a view to animals, isobiotic animals in a moment. So if we look at the timeline of isogenic mice, and this I'm going to contrast with isobiotic mice in a moment, it has only a few of the annotations of the people who've made massive contributions to this. But you can see that from the start of the Granby Farms and Abbey Lathrop in the 1900s and the development of the Jackson Laboratory, we now have enormous numbers of embryonic stem cells, live mice, frozen embryos, and frozen sperm. And these are generally very widely shared amongst biomedical and biological researchers and provide the kind of impetus that has driven a huge advance in understanding of live in vivo modeling in our careers. So the standardization that can be achieved, of course, depends not just on the standardization of genetics. So this shows one of many sorts of experiments of taking two inbred strains in an F1 hybrid and showing that the kidney weights here are closer together, have a lower standard deviation compared with the F2 hybrid or outbred. It makes another important point that being free of pathogens also reduces the standard deviation. Now, the difficulty is how one deals with this, given the galaxies of microbiota that are present in every vivarium and are, on the whole, different from vivarium to vivarium. And Tad Stappenbeck and Skip Virgin published a review in Nature earlier this year where they made the point that if you are to distinguish between genetic differences between two strains that are being bred separately and differences according to the microbiota, what you need to do is to breed Littermate controls, test for phenotypic differences, and then to know whether within those Littermate controls the mutants are not equal to the controls, which favors the fact that there is a genetic, host genetics underlying it, or the mutants are the same as controls, which favors the microbiome trait dominance. And this, I think, is a very important factor to consider in experiments, and it potentially can be done in all vivaria. The difficulty with all our vivaria is that in reducing pathogens, we also change the phenotype of the mice. And this is another paper that was published earlier this year that looks at the pet shop mice. And what one can see from these flow cytometric plots, looking either at peripheral blood cells or cervical tissue, is that one has a rather immature immunological state in the laboratory mice with rather little evidence of persistent antigen stimulation that's comparable to the neonatal human, but is not comparable to the adult human or the feral mouse. This, of course, is another difficulty of the sort of systems of notobiology that I'm about to describe, that even here in a SPF situation with a diverse microbiota, comparatively speaking, one is dealing with a rather limited immunological phenotype. is it possible to inbreed mice in the sort of way that you would do in order to create an inbred strain and standardize the microbiota? And this has been tested out, and of course it doesn't work properly. So here, this is this is a example of a publication where they attempted to do this with Balb-C mice, and essentially they were not able to achieve any greater microbiota similarity as they continued the inbreeding of parentals that were selected to have very similar microbiota similarity. So this is not an effective way of going forwards. So what are the options? Well, one is to allow the natural process to transfer a microbiota. And this can actually be done in a controlled way, for example, with an embryo transfer, so that embryos of a particular genetic strain are transferred into a strain that has the required microbiota, and then the maternal microbiota of the mother will be acquired. Another approach is to, is to gavage with organisms from a pure culture, feces, or add a colonized animal to a cage. This, for example, can be a method of fecal transplantation, or one can actually carry out a specific colonization with defined culturable organisms in order to create an isobiotic mouse. I shall come to this in a moment. The third way is to use a reversible system to gavage with bacteria, for example, that cannot persist within the mouse so that the mouse becomes germ-free again. And so you uncouple the effect of exposure to live microbes from permanent colonization, which allows one quite powerfully to dissect the underlying mechanisms. So notobiotic microbiotas, standardized microbiotas, were actually, if you look back at the literature, initially devised in order to try and avoid fatal opportunistic infections in germ-free mice. That was the original goal. And actually compared with the isogenic models, which I've been shown have been developed in enormous numbers, relatively little effort has been made to have standardized, stable microbiotas that are representative of diversely colonized animals for at least some traits. So if you look at the isobiotic timeline, you find that, again, I haven't put every contribution on this, but it is notably sparser than the isogenic timeline in terms of the development of standardized notobiotic microbiotas. And we have relatively few that can be shared across different vivaria. But it is possible to use the power of the isogenic animals by transferring the microbiota through an embryo transfer procedure from a strain with the desired microbiota. So to take the embryos from an undefined microbiota and put it into a pseudo-pregnant female, and then the pups of the isogenic strain will be born with the required isobiotic microbiota. So this at least is a handleable situation. So if I first just show you reversible colonization, this was developed by Siggy Hapfelmeyer when he was in my laboratory, and it depends on the fact that the E. coli organism is being deleted for D-alanine synthesis and diaminopymelic acid. Of course, it's L-alanine that is in the eukaryotic peptides and proteins. So this is a bacterial-specific amino acid. And if you look at the blue circles of an inoculation of a germ-free mouse, you can see that there is a wave of shedding of live microbes but the animal becomes germ-free again, because without the ability to cross-link the peptidoglycan, it cannot persist, and these auxotrophic requirements are not available within the intestine. Whereas in the filled circles, you can see that there is, this is the parent strain, there is a permanent colonization. So this has been used in a number of different models. It will nicely induce immunity. This is something that we have done more recently to do transient colonization of a pregnant dam, so that the pregnancy ends with the mother and pups germ-free. They're born germ-free, and then the germ-free pups are analyzed according to whether they came from a control mother that was germ-free throughout, or whether it comes from a mother that was transiently colonized. And this has a remarkable effect on the neonatal immune system of the pups that are themselves always germ-free. So there is, if the mother was transiently colonized, there is a change in the transcriptional profile of the epithelial cells with antimicrobial peptides and mucus. There is a change in the populations of the innate immune system. All these are absent in the pups born to the germ-free mother throughout, with functional effects on the penetration of bacteria, translocation through the epithelial surface. Another nice thing about this sort of system is that it allows one to actually follow molecules, and part of this effect is due to bacterial molecules that are ligands for the arrow hydrocarbon receptor. I haven't got time to go further into that. I'd like to move on to looking at more diverse consortia. Now, the best known of these, of course, is the altered Schaedler flora that is distributed. It often is diversified in Vivaria, at least those that we have looked at. And it was originally isolated, devised by isolations from Russell Schaedler and Roger Orcott, and here are the metadata. So if one uses this strictly, as I told you, you can transfer this onto different strains by embryo transfer. And this is an experiment looking, is just an example experiment done by Marcus Goiking, looking at the induction of regulatory T cells with flow cytometry in the lamina propria. And you can see that even though this is a very benign microbiota, it induces regulatory T cells, and these are Helios low, so they are inducible regulatory T cells. But the point is that one can move across different backgrounds and look at the microbiota for a particular readout across different isogenic strains. So the approach of generating isobiotic lines is to use defined mouse or human commensals, and then to inoculate them. And of course, this depends on what actually will take within the mouse intestine and what turns out to be stable. So the object is to try and have as complementary a metabolic set of pathways as possible, and if possible to try and have something that metabolomically and in terms of the animal disease models is reflective of a more diverse microbiota. And with this, of course, then you can make differences and potentially, just as one can have isogenic mice that have plus or minus one gene, so you can have conbiotic microbiotas as well. So the criteria that I would like to propose for isobiotic strains are, ideally, they should be stable over generations on open source diets. I think it's very important that for reproducibility, we have as little magic and as little patent restriction as possible. That the members are culturable with published genomic sequences and open source stocks, so other people can potentially regenerate the microbiota, and one has a way of dealing with drift. Is transferable to different genetic backgrounds and to different institutional vivaria. It can be regenerated from pure culture. There is a completeness of the metabolic pathways. There are no abnormalities in the essential clinical chemistry, histology, or fecundity. And there is some representative nature of metabolic and immunological profiles and pathogen resistance. Ideally, it would be relatively stable with aseptic husbandry in isolated ventilated cages, so that it isn't necessary to have germ-free standards of husbandry in order to be able to use these. So one that we have devised has been the SDMDM2. This shows the metadata. These organisms were isolated by Bärbel-Stecker in Munich, and the reconstitution was done by my colleague Kathy McCoy in Bern. They are fully sequenced. The sequences are published. They are orderable, so the pure stocks are orderable. And we have published a few things using this consortium. Final slide is just to make the point that there is actually, so it has an improved resistance to pathogens. This is data from Bärbel's laboratory looking at the altered Schädler flora that is actually given a control, so another altered Schädler flora, or this SDM microbiota, or a diverse microbiota from pathogen-free mice, and then challenged with Salmonella typhimurium, an avirulent strain, and looking at the shedding. And you can see that although it doesn't quite reach the range of the controls, there is definitely colonization resistance that is not present in the altered Schädler flora. Now, I don't want to try and be prescriptive. I was discussing that with Skip Virgin before I started this talk. One microbiota doesn't, one swallow doesn't make a summer. One microbiota is insufficient for a scientific community. And yet it is possible to go some way further in terms of being able to have something that can actually read out the traits that we're interested in, but without having quite such anarchy, if you like, of having different microbiotas between different institutions. So the end and the challenges, as I see it, is that it is possible to create a variety of isogenic microbiotas with reasonable stability over time. And all science is an approximation to the truth, and this is just an approximation to a particular piece of truth. They can be transferred between institutions. They are powerful tools for exploring the consortial arrangements, and it also allows experimentation with small group numbers. I mean, it's enormously expensive doing these metagenomic, metatranscriptomic studies if one has to amplify the group numbers, and small group numbers of 3 or 4 animals are sufficient for this. There is inevitable genetic drift. As I've shown you, the diet must be controlled. There is a capability of disease modeling. And we argue that these should be freely available across different institutions as isogenic animal models, allowing reproducibility in different vivaria, or at least dissecting the reasons for the lack of reproducibility, if that is the case. And the vision is that these sorts of models should be as diverse and as freely available as the isogenic models are today. And finally, no one isobiotic model should be considered exclusive. I don't want to be prescriptive. It is possible with isogenic strains to take a factorial approach, so different isogenic strains model particular situations better than other isogenic strains. And this is a more powerful statistical way to proceed, and because it involves fewer animal numbers, is also more ethical, I would submit. And I feel that we could take a similar factorial approach with different notobiotic standardized microbiotas. So at the end, I'd like to make acknowledgements. This is an incomplete list. Bärbel Stecker, with whom we've collaborated, as I said. My colleague, Kathy McCoy, we did this work in Bern. She's now at the University of Calgary in Canada. And colleagues at the ETH in Zurich, Uwe Zoyer and Toby Führer. And this talk, of course, would be totally incomplete without thanking the staff of the different animal facilities involved and all of you for your attention.